This is the first section of our lecture. In this section, we are going to talk about the deficiencies in secretions and function. Let me give you an overview that we are talking about the neurohypophysis or posterior lobe of the pituitary gland, which is located in the sella turcica of the sphenoid bone, and it produces two main hormones, AVP, also known as antidiuretic hormone, and oxytocin. And these two hormones are basically produced from the axons in the hypothalamus, whose cell bodies are present in the hypothalamus, and they are then transported down the axons into the posterior pituitary, and where they are released into circulation in response to specific stimuli. Moving on to our first section, we are going to talk about the disorders of neurohypophysis. We will discuss AVP and oxytocin in detail, and what are the deficiencies in AVP secretion and function. Now, this is the diagrammatic representation or the anatomy of neurohypophysis or the hypothalamic hypophyseal system. This is the exact location in the brain where pituitary gland is present, and this is a magnified view of this pituitary gland. So as you can see here, the antidiuretic hormone or AVP is released from the neurosecretory cells of the paraventricular nucleus of hypothalamus, and oxytocin is released from the neurosecretory cells of supraoptic nucleus. Both of these hormones are then transported down the axons into the posterior pituitary through the infundibulum, in which lies the axonal tracts. And in the posterior pituitary, these hormones are stored in cells, and they are released into the circulation in response to specific stimuli. So this is the complete diagram of the hypothalamic hypophyseal system. This is anterior pituitary, which is glandular in origin, as compared to the posterior pituitary, which is composed of largely axons. Now we will talk about ADH. ADH is basically a naturally occurring antidiuretic hormone in humans. It is also known as arginine vasopressin. It's basically a nonapeptide. That means it contains 9 amino acids, which are divided into a 6-member disulfide ring and a tripeptide tail. There is a variant of arginine vasopressin, that is lysine vasopressin. It has a lysine in place of arginine as the 8th amino acid, as you can see in this diagram here. Where in arginine vasopressin, both of their structures are similar, but in AVP there is arginine amino acid in the 8th position, and in lysine there is lysine amino acid in the 8th position. This lysine vasopressin is not found in humans. It is found in pigs and some related animals. Basically, ADH is synthesized via a polypeptide precursor in the neurosecretory cells of the hypothalamus. It includes AVP, neurophysin, and copeptin. All of these are encoded on the chromosome 20. After the preliminary processing and folding of these polypeptides, these precursors or the final products are packaged into neurosecretory vesicles, and they are then transported down the axon through the infundibulum, and they are further processed to AVP, neurophysin, and copeptin. After the complete processing, they are stored in the neurosecretory vesicles until they are released by certain specific stimuli. And they are released by the process of exocytosis into peripheral blood or capillaries, which are present in the posterior pituitary. Now, if we talk about the receptors of ADH, there are 3 main receptors on which ADH acts. These are V1A receptors. These receptors are basically found on the arteries, and they are responsible for vasoconstriction. Then we have the V1B receptors. These receptors are found on the adrenal glands and they are responsible for the stimulation of ACTH secretion. And then there are V2 receptors. These receptors are responsible for the insertion of water channels, aquaporin 2 channels, in the luminal membrane of collecting ducts. These receptors are found in the kidney, and they are encoded by the chromosome 12q13. If we talk about the regulation of ADH, ADH is mainly regulated by the effective osmotic pressure of body fluids. How these osmotic pressures are regulated by the body? They are mediated by specialized hypothalamic cells, which are called as osmoreceptors. These osmoreceptors are extremely sensitive to small changes in plasma concentration of sodium and its anions, whereas they are insensitive to other solutes such as urea and glucose. So they basically respond to the plasma concentrations of sodium and its anions. Now, osmoreceptors, they contain both the inhibitory as well as the stimulatory components to form a threshold. And that threshold is very important. What happens below the threshold is that plasma AVP is suppressed to allow maximum water diuresis. If the osmoreceptors are not reaching that particular threshold level, the secretion of AVP will be stopped. In case of if the osmoreceptors have crossed the threshold, plasma AVP will start rising steeply in direct proportion to the plasma osmolarity. So plasma osmolarity is one of the main factors in of regulation of ADH. Now, what is the threshold for AVP release? We said that basically ADH release is mainly dependent on the plasma concentrations of sodium and its anions and on the plasma osmolarity. So what are the values of plasma osmolarity and plasma sodium? These are plasma osmolarity of 280 milliosmoles per liter and above, and sodium 135 milliequivalents per liter or above is responsible for the regulation of ADH or for the release of AVP. Now, there are certain set points of osmoregulatory system that can be lowered by, or in other words, you can say that there are certain points through which we can lower the threshold of osmoreceptors. What are those set points? They can be lowered by the causes. These are pregnancy, menstrual cycle, estrogen use, and whenever there are large acute reductions in blood pressure or volume, as that occurs in a hemorrhage, or if the patient is in shock, all these cases are responsible for lowering the threshold of osmoregulatory system. Now, how are these osmoregulatory system or these osmoreceptors responsible for regulating ADH? What happens in the process? These are basically mediated by neuronal afferents. These neuronal afferents originate in the transmural pressure receptors, and they are found in heart and large arteries. They we also they are also found in the carotid bodies, and through these carotid bodies, they project via the vagus and glossopharyngeal nerves to the brainstem. These nerves are also called the herring nerves. These postsynaptic projections, they ascend to the hypothalamus. They maintain a tonic inhibitory tone, and what happens is this tone is decreased when the blood volume or pressure falls by more than 10 to 20%. Okay. So what is happening in this process? Whenever there is large acute reductions in blood pressure or volume in the body, these osmoreceptors or these carotid body receptors, they will not detect that tone which is normally maintained by these receptors. When they will not detect that tone, they will send a signal through the glossopharyngeal or vagus nerves to the hypothalamus, and hypothalamus will detect these changes and it will start releasing the increase in release of AVP, and so that these pressures can be brought back to normal levels. Now, the AVP secretion is stimulated by certain other factors. These include nausea, there is acute hypoglycemia, there is glucocorticoid deficiency, smoking, and hyperangiotensinemia. These factors are also responsible for the stimulation of AVP, along with the other changes we have discussed in the previous slide. Out of all these factors, nausea is the most important factor in the stimulation of AVP secretion. Now, if we talk about nausea, the emetic stimuli are extremely potent. They elicit immediate 50 to 100-fold increase in the plasma AVP. And whenever the nausea is transient and it is not associated with vomiting or other symptoms, it can still cause an immense increase in the secretion of AVP. They mainly act on the medulla through the emetic centers. And how we can block these stimulatory factors, or how we can control the nausea, we can give antiemetics to patients such as fluphenazine that can control this stimulatory factor, or it can stop the release of AVP. Pain or other noxious stresses, they have no direct effect on AVP. They only elicit a vasovagal reaction with nausea and hypertension if they are associated with nausea, because in all the cases pain is basically not responsible, or it is not acting on the hypothalamus. It's always the nausea that is going to act. So if pain is not associated with a vasovagal reaction, it is not going to produce any changes, or it is not going to stimulate any release of ADH. Now, if we talk about the action of ADH, as you can see, this is the diagram of a nephron. In a typical 70 kg adult, kidney normally filters around 180 liters of plasma per day. Out of this 180 liters, 144 liters, that is 80%, is reabsorbed isoosmotically in the proximal tubule, and the remaining 8 liters, that is 4 to 5%, it is reabsorbed without solute in descending limb of the Henle's loop. Now, what happens to the remaining? The remainder is diluted to an osmolarity of 60 millimoles per liter, 60 millimoles per kg. How it is diluted? By selective reabsorption of sodium and chloride in the ascending limb of Henle. In the absence of AVP, urine that is issuing from the loop, it passes largely unmodified. It passes largely unmodified through the distal tubules and collecting ducts, and it results in a maximum water diuresis. So there will be no absorption of water in the absence of AVP. Whereas in presence of AVP, solute-free water is reabsorbed osmotically through principal cells of the collecting ducts over here, resulting in excretion of a much smaller volume of concentrated urine. So there will not be secretion of the excessive dilute urine. It will be concentrated and it will be reabsorbed in the body over here in the collecting tubules. This antidiuretic effect, now we see what is the mechanism going behind this effect. This antidiuretic effect is mediated via a G protein coupled V2 receptor. Now this G protein coupled V2 receptor, it increases the intracellular cyclic AMP. What happens? This intracellular cyclic AMP increases the translocation of aquaporin 2 water channels into the apical membrane. Now, this is the apical membrane and this is the basal membrane. So when AVP acts on the V2 receptors, it increases the intracellular cyclic AMP. This cyclic AMP acts on the aquaporin 2 water channels, and these aquaporin water channels get inserted into the apical membrane. The resultant increase in permeability, it permits an influx of water that diffuses out of cells through aquaporin 3 and aquaporin 4 water channels on the basal lateral surface over here. Now, the net rate of flux across the cell is determined by the number of aquaporin 2 water channels in apical membrane and the strength of osmotic gradient between tubular fluid and the renal medulla. Tight junctions that are present on the lateral surface of cells. What is their function? They serve to prevent the unregulated water flow. So this is basically the main action of ADH. ADH basically reduces water excretion by promoting the concentration of urine. How it is done? It is achieved by increasing the hydroosmotic permeability of cells. Now, these cells which are lining the distal tubule and medullary collecting ducts of the kidney, in the absence of AVP, these cells are impermeable to water, as I have already discussed in the previous diagram. But when AVP acts on these cells, there is insertion of aquaporin 2 water channels and they will become permeable to water and resulting in maximum water diuresis. These cells, in the absence of AVP, they reabsorb little of the relatively large volume of dilute filtrate that is entering the proximal part of the nephron. It will excrete very large volumes, 0.2 ml per kg per minute of the maximally dilute urine. What happens to the specific gravity? It will also increase, it will be 1.00, and the osmolarity of urine will be around 50 milliosmoles per liter because there is maximum water diuresis. Whereas the osmolarity of plasma will start increasing because there is maximum reabsorption of the solute. Now, the magnitude with which the water diuresis is occurring, it varies in direct proportion to plasma AVP concentration. And the second thing is the rate of solute excretion. These two things are determining the magnitude. At maximum levels of AVP and normal rates of solute excretion, it approximates around the urine flow rate will be as low as 0.35 ml per minute, whereas the urine osmolarity will be as high as 1200 milliosmoles at maximum levels of AVP. It will be reduced by solute diuresis, which occurs in diabetic patients because there is excretion of glucose in the urine. So because of this solute diuresis, the urine flow rate and the urine osmolarity will be decreased in case of diabetic patients. Now, how is antidiuresis mediated? It is mainly controlled by the G protein coupled V2 receptors on serosal surface of cell that occurs by the activation of adenyl cyclase. Whenever there is activation of adenyl cyclase, there will be insertion of aquaporin 2 water channels into the luminal surface, and V2 receptors and aquaporin 2. These are encoded by the genes which are found on chromosomes XQ28 and 12q13. Now, at high concentrations of AVP, it has certain different actions. It causes a contraction of smooth muscles of the blood vessels in skin and GI tract. It induces glycogenolysis in the liver, and it potentiates adrenocorticotropic hormone ACTH production that is released by the CRF or corticotropin releasing factor, and all this is mediated by V1A or V1B receptors that are coupled to phospholipase C. Now we will talk about the metabolism of AVP. It distributes rapidly into space, almost equal to the ECF volume, and it is cleared irreversibly with a half-life of 10 to 30 minutes. What happens during pregnancy? The metabolic clearance of AVP is increased 3 to 4-fold. That is why the set point of osmoregulatory system or the threshold is decreased in case of pregnancy. Thirst ensures adequate intake to prevent dehydration. It is regulated by an osmostat which is found in the anterior hypothalamus. It detects very small changes in plasma concentration of sodium and its anions, similar to the osmoreceptors that detect changes in plasma concentration of sodium and its anions. Osmostat is set about 3% higher than AVP osmostat. Now we will see what this was all about the physiology and the anatomy or the structure of AVP. Now we will see the structure of oxytocin. It is also a nonapeptide containing of 9 amino acid. It differs from AVP only at positions 3 and 8. Oxytocin has a little antidiuretic effect as compared to AVP, and it mainly acts on mammary ducts to facilitate milk letdown during nursing. The other function of oxytocin is that it helps initiate or facilitate labor by stimulating the contraction of uterine smooth muscles. However, there are no changes in the circulating levels of oxytocin in blood, but it does seem to act and help initiate labor. And there is another function. How is oxytocin regulated? It is regulated by suckling and dilatation of the cervix. Now, this is the diagrammatic representation of these paraventricular and supraoptic nuclei of hypothalamus. This is hypothalamus, and this is the infundibulum, and here we have pituitary gland. This is the anterior pituitary and the posterior pituitary. These arrows, these are basically the main pathways and the effects of AVP and oxytocin. Now, oxytocin and AVP are synthesized in the large magnocellular and small parvocellular neurons of the paraventricular and the supraoptic nucleus. AVP is also expressed in neurons in the suprachiasmatic nucleus, the central nucleus of amygdala, the bed nucleus of stria terminalis, and other regions. The extra hypothalamic expression of oxytocin is in generally similar to that of AVP. Now the axons of magnocellular paraventricular or the supraoptic nuclei, they synthesize AVP or oxytocin. They project to the posterior pituitary, from where these peptides are released into general circulation. The circulating AVP has antidiuretic and vasoconstrictor effects, whereas we have already discussed that oxytocin has a major role in parturition and lactation. Axons of parvocellular oxytocin and AVP neurons, they are distributed in several areas of CNS. Now, what are their function in other areas? These neuropeptides, they modulate several functions that include the social behavior of a person by acting on the lateral septal nuclei. They also control the behavioral emotional responses, the cardiovascular functions such as the sympathetic activation and the parasympathetic activation. nociception or antinociception by acting on the dorsal horn, and the motor neuron excitability by acting on the ventral horn. Now, AVP that is released from the suprachiasmatic nucleus, it contributes to the control of circadian rhythms and body temperature. How it does that? It acts via projections to medial preoptic area and it by medial preoptic area and the subparaventricular zone and the dorsomedial hypothalamic nuclei. So the other functions these hormones are performing are they are also controlling the social behavior. They are controlling the circadian rhythms by acting on the suprachiasmatic nuclei. They are also controlling the body temperature of a human body. They are acting on the brainstem and spinal cord, and they are controlling the sympathetic activation. They are through the autonomic nuclei. They are controlling the parasympathetic activation. By acting on the dorsal horns and the ventral horns, they are controlling antinociception and the motor neuron excitability. So these are the other pathways through which AVP and oxytocin are causing symptoms in the body or controlling certain actions of the body. Now, coming to the deficiencies of AVP secretion and action. Whenever there is inappropriate or deficient secretion of AVP, there will be a disease called diabetes insipidus that is characterized by production of large volumes of dilute urine. Whereas in case of the excessive production of AVP, there will be a disease known as hypodipsic hyponatremia or hyponatremia due to inappropriate antidiuresis or SIADH. We will discuss these diseases or these syndromes in our subsequent sections. This was all about our first section. Keep watching sqadia.com.